Swayam Prabha. Digital India. Educated India.
Welcome back to the organ printing course. So in this lecture, we'll discuss bioprinting of different target organs, and so we'll see few of the organs how bioprinting technology can be used for fabricating that particular target tissue or organs. So first thing we'll discuss about the bioprinting of vasculature. As I have already mentioned in my earlier lectures, that vasculature is the most important thing for sustaining the growth and function of different target tissues and organs. So very first challenge to fabricate a solid organs is the fabrication of vasculature or embedded vasculature system within that bioprinted organ. So we'll discuss what are the different strategies. So first we'll discuss what is the anatomy of blood vessels, different blood vessels hierarchy, and what different types of different modalities of bioprinting can be used for developing this bioprinted vascularized tissue construct, and also we'll discuss what are the different strategies in that domain. So let us first understand what is the need for developing vasculature within a bioprinted construct. As we have already earlier discussed, that blood vessels provide nutrition, oxygen to all the tissue, all the cells present in that particular tissue or organ, and also it helps in removal of waste material generated by the cellular metabolism to the and then bring that to the kidney or other excretory organ for excretion purpose. So vascular tissue is the driving force for organ growth and function. So we need to definitely so we need to provide that vascular tissue to the bioprinted tissue or organs. And this vascular need for vascularity is not so drastic for flat and tubular organs like skin, airway tract like trachea and other tissues, because in those organs, those organs they are mostly flat and thickness is not that great. Like in some certain cases, the thickness reaches to a probably a few mm or so. So in that case, the nutrition oxygen can diffuse from the nearby vessel to the tissue by can diffuse from the nearby vessels, but and the cells in that those tissues can sustain and survive. But for solid organs and also for hollow organs like bladder and solid organs like kidney, liver, pancreas, and others, there the blood vessel is very much required. The blood vessel supply or the blood vascularity or the vascular tree is very much required because the wall thickness there exceeds 300 micrometer. So that is most important. So the important thing to remember here is for hollow and solid organs, vasculature need to be developed, or vascular tree has to be bioprinted along with the bioprinting of that particular organ. But it is not so drastic for the flat and tubular organs. And there have been few attempts to promote infiltration of blood vessels from the capillary network within the blood printed construct. So that is another strategy few of people researchers have been working on, how to induce the infiltration of blood vessels from the existing blood vessels, that is, or how the sprouting or the angiogenesis can happen, and that can so the blood vascular tree of our blood or blood vascular tissue growth can vascular tissue can ingrow within the infiltrate within the tissue. So there are few attempts to do that because replication of the hierarchical vascular network is very challenging, but that is very much required for biofabrication of complex tissues. Okay. Because the blood vessels, there is a particular hierarchy, and that hierarchy has to be maintained within a within the biofabricated tissues and organs, so that all the cells present in part of different parts of that particular tissue organ can get nutrition or oxygen. So, for whole organ engineering, vascular bioprinting is very much important, and that is the need. Otherwise, the whole organ cannot sustain and the function cannot be fully established. Let us now understand the vascular anatomy. Before actually even targeting the vascular bioprinting, we need to understand what is the different structure present there, what are the different what are the different structure, and the dimensions of those structure is very important to understand, because each of these different structure can be targeted by one or other bioprinting modalities. So the blood vessels is mainly composed of three concentric layers. Like this blood vessel, the tubular structure, that is composed of three layers, where the cells, different cells and extracellular matrix are organized into and develop that kind of tissue structures. Like tunica intima is the innermost layer of that blood vessels, that is composed of endothelial cells, where the endothelial cells they form a layer in the innermost part of that tubular structure. So the tubular structure, so that is tunica intima. The next layer, that is the muscular layer, that is present just above or above the endothelial layer, that is composed of the muscle layer, muscular tissue. So where that is made, so that's the tunica in media, so where mostly smooth muscle cells are present. And then the next, on top of that, there is the outermost layer is the tunica externa, that is the epithelial there, the where the epithelial cells are present, and also other type of cells are present. So these are the main three layers of bigger blood vessels. Now then, the artery, or the from the heart, the aorta generated, and that aorta again branch into arteries, and then that artery again branch into microvasculature. When they actually this micro then artery that reaches the tissue bed or the organ bed, then they form different types of smaller microstructures, like arteriole, capillaries, venules, all these things are formed there. And they are then these small or these microstructures, they extend the blood supply to the whole organs. Okay. The arterioles have a diameter of almost like 10 to 200 micron, and the arterioles also they retain all this tunica layers, like intima, media, and externa. All three layers, tunica layer, these three layers are present in the arterioles. Arterioles again branch into capillaries in the tissue bed or organ bed, where the lumen diameter can reach up to that very fine structure, like 5 to 10 micron of capillaries are possible in capillaries exist in the tissue or organ bed. And these capillaries, they retain only the endothelial layer and a basement membrane. So the capillary, actually all three layers like this tunica intima, tunica media, or tunica externa are not present. They are the endothelial layer present, and on top of that the basement membrane, and on top of that the pericyte cells also attached to that basement membrane. And then they actually the function of the pericyte, they stabilize the vessel, and also it allows the diffusion of solids from to the nearby cells and tissues. Solids tissue. So the in the base, the important thing to understand here is the capillaries. Suppose we are targeting capillaries, that the capillaries composed of intima layer, that is the endothelial, and then on top of that there's a very fine layer of basement membrane and the pericytes there. So other structures are not present here. So the again, depending upon the structure of the basement membrane and also the intercellular spaces, again these capillaries can also lead to results in different types of structures, like either it can be a continuous capillary or it can be fenestrated or sinusoids, and the different say different cells or different organs have different kind of capillary structures, like the continuous capillaries where the basement membrane is mostly continuous. There it is present in skin, muscle, lung, and central nervous system, where the basement membrane is continuous, and so this kind of continuous capillaries are present. So because, and please remember this, when the basement membrane is intact, it actually modulate the diffusion of solids to that particular tissue, surrounding tissue or cells. So in case of continuous, there is a but definitely there is a more typical modulation of this the diffusion of these solids. Then the second category is fenestrated capillaries. There, where the basement membrane is not continuous, there can be some kind of fenestration. It is present in in renal glomeruli, intestinal mucosa, and exocrine glands. When that there, the diffusion is very different than the continuous continuous capillaries. Then the other class is sinusoidal capillaries, where the intercellular gap is larger, and it is mostly present in liver, spleen, and bone marrow. And the sinusoid, these are the three types of capillaries. And if you are targeting a particular tissue structure or tissue organ structure, then you have to also understand what type of capillary is actually present in that kind of tissue or organ, so that actually you can mimic that particular capillary structure by bioprinting. The next thing is the vein and the venules, where the in the tissue from the tissue bed, when the capillaries again they reunite and they form the venules, so that they empty all the bloods in the venules. So the venules are typically, say, starting from 8 to 800 microns of lumen diameter, and this they carry the after the capillary, they carry the blood to the blood comes to the venules. From venules again it comes to the much larger veins. But again the venules, where again all three layers like externa and a thin medial layer and the endothelium is present are present in case of venules. And again the multiple venules they empty into the larger veins, like, and larger veins also all three layers, all the three tunica layers are present. And again larger veins, what they do, they transport back the blood to the heart for pulmonary circulation. So the blood starting from the heart, blood travels by aorta to artery to arteriole to capillary to venous to veins and to vena cava, larger veins, and again to vena cava. Through that, the the whole blood is circulated with the pulmonary circulation, for pulmonary circulation and for further systemic circulation. Now, blood vessels hierarchy. This is the blood vessel hierarchy, where on what kind of bioprinting modalities can be employed for this. We need us understand that. As I already mentioned, aorta diameter of aorta is like 1 to 2 centimeter. So where mostly bioprinting modalities are not being used, and molding is technique is the most favored one, because this large diameter aorta with bioprinting, it is challenging to make, but molding technique is very much very much suitable for this kind of generation of this aorta, where the diameter is more. Next is the artery, where the diameter is can be anywhere between 1 to 5 millimeter, where inkjet assisted and extrusion bioprinting is these two modalities can be very well used for generating these artery structures. Next is the arterioles. Arterioles are much finer structure, like starting from 50 to 100 micron, where inkjet assisted bioprinting and also laser assisted bioprinting can be employed to generate this kind of arterial structure, mostly because of their finite dimensions. Right? Then again, the next is the capillaries, which is the capillaries are present in the tissue bed or organ bed, where the diameter is very very fine, like starting from 10 to 30 micron. So the lumen diameter is the typical lumen diameter is like in the range of 20, around 20 micron. So that is that much fine architecture or cap structure can be developed by light assisted bioprinting, because we have discussed this light assisted bioprinting there. The resolution is very very high. So that kind of technique or laser assisted bioprinting, that kind of techniques can be very well used to degenerate this capillary structure having dimension of 10 to 30 micron. The next is the venules, where as I discussed earlier, where the capillaries reunite and then they drain the blood to the venules. So venules have a typical diameter of 30 to 50 micron lumen diameter. So this also can be very well printed with laser assisted or light assisted bioprinting because of this high resolution thing. And then the veins where the diameter is typically zero point 0.1 to 1 mm, struck 1 mm. So this can be very well printed, again printed with inkjet assisted and extrusion based bioprinting techniques. And then the next thing is the vena cava, where the diameter typically from zero point 0.5 to 1 centimeter, and it is because it is a thick blood vessels, so it is also very well can be printed with molding technique, but other technique is not that suitable thing. So this is the horizon of all these different types of blood vessel structures, and these are the respective bioprinting modalities that can be very well used for targeting that particular or to develop that particular blood vessel structure. Now one thing is pretty common, or it is understood by after several fundamental studies, that vascular formation can govern the tissue formation. So suppose if the blood vessels a vascular tissue formation, if you take care of the vascular tissue formation within a bioprinted construct, then that tissue or that particular bioprinted tissue or organ, that formation, their maturation formation can happen simultaneously. Now, the most vital organs that the current researchers are working on, like liver, lung, kidney, pancreas, and brain. So these are the solid organs. Those are now people now various researchers are targeting. And if you see these type of solid organs, they are highly vascularized. So because they are highly vascular, because these organs are also metabolically very active. So they need lot of oxygen nutrition for their survival and also for their function. So that's why these organs are highly vascularized. And then they, so for bioprinting that kind of thing, they require vascular network with high fidelity and functionality, because the blood vessels we need to replicate the that kind of vascular, that kind of blood vessel formation, that vascular tissue formation with high fidelity. So that structure, as I mentioned on the last slide, that the structure, that the dimension of all these blood vessels, we need to develop or need to print that kind of vascular network with those dimensions. Typically strict to those dimensions we have to print, and then only that function can be established, and then that tissue can be vascularized. Another most important thing is that if you see solid organs, they are the vascular tissue is present throughout that solid organ. That means each and every cells in that blood, in that particular tissue, getting nutrition, oxygen from the nearby blood vessels. This is mostly the capillary structure that is present in the blood within that tissue bed. So all this, so that solid organ should be highly vascularized. And when we are targeting to print a solid organ, then also we need to take care of that vascular tree. That vascular tree has to be printed, and then that can be then only that particular solid organ can be developed. Why it is important? Because if you see, it has been found over by 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 the various researchers over experiments that the cells cannot survive if that particular cell is present beyond 300 micrometer distance from an existing blood vessels or typically a capillary structures, because that nutrition or oxygen cannot reach beyond that limit. So that's why the cells cannot survive. So that is the one. So that's why when we design a vascular tree within a bioprinted tissue or organ, we need to remember that all cells present in the tissue or organ bed should be present within that distance from the nearby vessel. So that is the limit. That is the limit, and we have to always remember that. Now, another important thing is innervation in that bioprinted construct, and it has been found that innervation along with blood vessel formation can promote the integration of these engineered organs with the pre-existing organs. Like suppose there is a defect in an organ and we are putting a bioprinted organ, the bioprinted tissue organ there along with that, then that bioprinted organ can well integrate with the pre-existing part of that tissue or organ if the blood vessel formation can happen, if the innervation can happen, that lead to further functional tissue formation. Now there are for larger blood vessels, synthetic grafts are very much popular for these larger blood vessels, and there are various success stories also. This larger blood vessels mostly made up of either extended PTFE or by other material. Those like grafts are very much popular and successful also because they can replicate the vascular tissue formation within this thing, but they can very well integrate with the existing blood vessels. So this is very effective for large and medium large vessels, like that is where the blood vessel like size is more than 8 mm, but that is not so successful in case of small blood vessels, like where the diameter is less than 6 mm. So that is not very successful, such as that is present in case of coronary arteries, where the small blood vessel like less than 6 mm blood vessels are present, and that carries blood to the whole heart muscles. And there autologous grafting is the most preferred one, like for this all this interstitial thoracic or radial arteries, where autologous grafting is most preferred. Why it is challenging to bioprint small blood cells is the because one thing is due to lack of endothelial cells, because in smaller blood vessels again we need to infuse that endothelial cells within the smaller blood vessels, so the endothelial cells can go and bind to the inner luminal structure of that blood vessels, small blood vessels. There is a challenge. Again, the diameters we need to take care of the dimensions, where the diameter of the blood vessel is such that it is challenging to biofabricate that. Another thing is the compliance or the mechanical property can mismatch is another reason that is the challenge for this. And that is another reason why this smaller diameter blood vessels cannot, yeah, it's really challenging to bioprint. And most of the cases, these small diameter blood vessels printed or developed with the synthetic material, they fail in clinical setting because of the intimal hyperplasia or thrombosis or graft site infections. Because of any of these cases, and unless it's like I miss over tissue growth or there's a thrombosis, that means the blood clot within the graft, or double infection during the graft site. Because of all these reasons, smaller diameter blood vessels with synthetic smaller diameter blood vessel graft is not successful, and bioprinting can be one option by which the smaller diameter blood vessels can be very well developed. So as I already earlier mentioned that for large diameter blood vessels, uh, molding technique is preferred where synthetic graft can be very well manufactured with this molding technique, and that those are very much effective for larger diameter blood vessels. But for smaller diameter blood vessels, synthetic graft is not much successful, and there are graft failures due to various reasons. And because of all these things, FDA till now FDA has not approved any small caliber vascular graft where the vascular graft is less than 5 millimeter. So those kind of things are not approved by FDA, and those are not available in clinics. This thing with this. Now bioprinting, that's why bioprinting of smaller diameter arteries and veins are has been or have been the option. So has been the option for this. With bioprinting, we can definitely fabricate or those kind of small diameter and veins. So bioprinting of small diameter arteries and veins that's feasible. And here again we have to endothelize the luminal structure, the lumen diameter or the internal structures with the thing. And that is another challenging thing, but that has to be done, and that is very much required. Other than this thing is the challenging to replicate the delicate anatomy of the arterioles, capillaries, and the veins. If you see in the tissue organ bed, there the arterioles and capillaries, the structure is very delicate. The anatomy of this thing very delicate. That is reproducing that thing or replicating this thing is another challenging. And to tackle that, tissue engineers actually have employed different strategies where angiogenic remodeling and different biofabrication strategies have been employed. So in this, so we'll discuss few of these things, how what are the different strategies for angiogenic remodeling, and also for what are the different biofabrication strategies those are employed for bioprinting of small diameter arteries and veins. Let us first start with this. Understand the angiogenic remodeling. Angiogenic remodeling means angiogenesis is basically the definition of angiogenesis is sprouting of new blood vessels from the existing one. That can happen under the induction of different factors, like growth factor is one such thing by which that can happen. So two main strategies for angiogenic remodeling have been employed, like either providing growth factors so that the rapid capillary formation inside the implanted construct happens. Suppose when you biofabricate a construct, we can always load growth factors in that. That means we can load growth factors in our biowing, and then we print the whole structure, print the whole tissue. So that is loaded, that so that that tissue along with the cells, the growth factors will be loaded in that, and those growth factors can induce rapid capillary formation in the inside that construct. So this is one, this by this way also we can yeah, remodel the angiogenesis in the bioprinted tissue construct. Another thing is stimulating blood vessels infiltration by host vasculature. That means in the host, in the bioprinted constructs also, we can actually print blood vessels, and they are the new blood vessels can also the blood vessels can infiltrate from the host tissue. Also the blood vessels can infiltrate. By these two ways the angiogenic remodeling can happen, and there are actually various growth factors those are used for this rapid capillary formation within the thing. The growth factors like vascular endothelial growth factors, VGF, fibrous growth factor like FGF, and platelet derived growth factor PDGF, and transformative growth factor TGF. So these growth factors have been used to deliver along with within the biowing for developing preparing the biofabrication contract, either alone or in combination. The other thing, this is different different materials have been also been used to load this growth factor to generate different types of structures, so that the, the blood vessel formation can happen within the biofabrication construct. And it has been found that when these growth factors are loaded within this kind of within this within the biofabric construct, then the vascular tissue formation is much higher in case of when the growth factors are loaded, and that too if the growth factors that can be released in a sustained manner, that lead to higher amount of blood vessels infiltration or rapid capillary formation within the biofabrication construct. Other than that also, when the blood vessels, when the growth factors are also incorporated within this uh, biofabrication construct, they also induce the stromal cells present in the surrounding tissue to differentiate into endothelial cells and form the vascular network. So that is the another way by which that stimulating blood vessels from the host vasculature, it can be stimulated by the, so that the stromal cells can be recruited and then differentiate into blood vessels like endothelial cells, and then they can form the vascular network. Other than this, also the growth factors are in certain cases that can promote the recruitment of circulating stem cells through specific antibodies. So that is also another way to whereby which we can promote the circulating or recruit the circulating stem cells, and that is because the stem cells, there are certain circulating stem cells that is present within the blood, and that is getting circulated all the time. So that if there is certain factors that can promote the recruitment of those things, circulating stem cells, and then induce the uh, blood vessels or tissue form blood tissue sorry vascular tissue formation within the biofabrication constructs, that can be beneficial or that can be helpful for vascularized tissue construct development with bioprinting. So this is the strategy by which angiogenic remodeling can happen. So thank you all for attending. In the next lecture, we'll discuss how by different biofabrication strategies are employed for blood vascular tissue formation. Thank you very much.